Breaking news! Breaking news! Inside your cells, there's a hard-working structure known as the rough endoplasmic reticulum, or the RER. Think of it as the cell's factory, a complex network where proteins are processed, folded, and prepared for their critical roles throughout the cell. How do scientists even see something this small? Great question! They use two powerful microscopy techniques. First, there's phase contrast microscopy. This technique doesn't require any staining, so scientists can look at live cells and see the RER without altering it. Phase contrast makes it possible to see all the membranes in the cell, which means the RER can be spotted in relation to other structures, giving a broad view of its network. Then there's immunofluorescence microscopy, which takes a different approach. This method uses special antibodies that are designed to attach only to proteins specific to the RER. These antibodies glow under fluorescent light, making only the RER membranes light up as if they're highlighted on a map. This allows scientists to zero in on just the RER itself, giving a clear, isolated view. But here's where it gets interesting. The RER isn't just a static structure sitting around inside the cell. It's constantly changing shape and moving, adapting to whatever the cell needs. The RER can undergo fission and fusion, meaning it can split apart and then merge back together as necessary. This dynamic movement allows it to change its structure and even migrate to different parts of the cell. Almost like a living, flexible factory floor that rearranges itself based on what's being built. To help visualize this movement, scientists use color codes in fluorescence imaging. DNA in the cell is usually marked in blue. Peroxisomes, small organelles involved in metabolism, are shown in green, and the RER appears in red. With these colors, scientists can track the movement and interactions of these parts in real time, giving them insights into how the RER supports the cell's constant act.